I'm going to be talking to you about some new findings in my lab that make me excited, which is investigation of WDR36. This is a novel gene that we think is related to glaucoma. I do have commercial and intellectual disclosures, none of which are relevant to this talk, but I do want to highlight funding relationships with things such as the Research to Prevent Blindness, as well as the NIH and NEI. I'm putting all love and positivity out to that particular organization as we all progress through this scientific landscape. This is a conglomerate of largely vision researchers and clinician scientists of all types. So I will quickly go through some of the facts about primary open-angle glaucoma, or glaucoma as it's sort of grab bag called, and then identify some of the clinical translational gaps that I have found in my own experience. Most of the glaucomas, specifically the primary open-angle glaucoma, in patients is demonstrated as a slow and largely irreversible disease. We know that this particular disease entity has a strong association with elevation of intraocular pressure. And as we begin to try to uncover the landscape of primary open-angle glaucoma, we are also beginning to appreciate the role that genetics plays in how this disease presents, whether this disease presents, and the progression with which this disease presents. We know that globally, the loss of retinal ganglion cells contributes to vision loss, but then as we begin to uncover some of the genetics, we're learning that the mode and the speed at which those retinal ganglion cells are lost really does differ among different populations of patients that carry the same primary open-angle glaucoma disease, which to me is both scientifically fascinating, but clinically translational fascinating, because it, as I practice, uncovers, I've listed two, there's probably a basket of gaps in the knowledge and clinical translational landscape that are worth probing and investigating into. Like, perhaps we need a more precise risk assessment or tools to delineate disease progression. Who is going to progress faster? Who is going to progress slower? And as we begin to uncover the role that some of these gene loci play in progression of this disease, there generally is a lack of understanding of how these loci contribute to the degeneration of retinal ganglion cells, the vulnerability of retinal ganglion cells to disease and injury. And all of that is a critical gap that I'm interested in probing. Take the heterogeneous disease phenomena. I have listed two types of glaucoma that continues to question and query people in my own, in the glaucoma field, both clinically and scientifically. We've got primary open-angle glaucoma. This is the most prevalent open-angle glaucoma that we experience. It's a silent and progressive course. We know that in this particular entity, elevated IOP in an open angle secure the diagnosis. And as we begin to uncover some, this is not at all in completion, that there are trabecular meshwork related disease loci and the neurodegenerative genes that contribute to how this disease progresses. The more commonly investigated genes like OPTN or TBK1 have been highlighted, but I hope to convince you that WDR36, which was largely forgotten, is worth investigation as well in terms of its neurodegenerative phenotype. But if you look at normal tension glaucoma, this is damage that occurs at normal intraocular pressures. We think that vascular and perfusion factors may play a role in how this disease progresses, but we know that these are largely IOP independent mechanisms, which has limited our ability to identify gene loci that are associated with this particular entity when we use things like IOP and sometimes even advanced cup to disc ratio to associate gene loci with this particular disease entity. So at the bottom is a phenotype where these two things converge. This is one of my own patients. In A, you're seeing a timeline of where I began to see some of the more structural and functional diseases, structural and functional damage occur in this patient. I really like this because I saw the hemorrhage, and then I was able, because of time, to trace the damage from that hemorrhage with the entities we use in clinic. And so in B, you can see the presence of the disc hemorrhage. In C, you can see the OCT finding, which presented 4 years later. And then finally, the visual field finding that presented along with that hemorrhage. And so you can see that one of the limitations and gaps is that we really want to be able to diagnose this person and treat this person before they get more hemorrhages that can result in irreversible vision loss. But also that there are additional gaps in clinic that prevent us from detecting both structural and functional damage from primary open-angle glaucoma that limits not just diagnosis, but the generation of neuroprotection in the future. And so, as I promised, WDR36, I think, is going to be an interesting genetic mutation for which to unveil some of these structure function differences, as well as potentially uncover some mechanisms of the disease. This is a nucleolar protein. It's involved in the maturation of the 18S ribosomal RNA. What's clinically important is that mutations in this particular gene have been demonstrated, depending on the study, between 1.6 and 17% of patients with POAG. Interestingly enough, at a talk similar, this very same talk 2 years ago, I was beginning to uncover some of this gene mutation, and I got introduced to Dr. Shefali Verma, who's a bioinformatician right here at the University of Penn, who was able to demonstrate and show that this is actually enriched in as a potential risk allele in the Penn Medicine Biobank and the POAG group study cases. So that was very interesting. It was an interesting collaboration that came about in a forum very much, this very same forum 2 years ago. And what we do know is that there are more severe phenotypes associated with this particular gene of certain mutations. Around the same time, I had a patient that had a particular type of this mutation, which probed the question further. And I just want to highlight, because I'm going to introduce a mouse model, which is subtly different than the substitution that we see in human disease, the substitutions we see in human disease. I will be studying a mouse model where there are 3 amino acids deleted in the functional pocket of this particular protein, whereas the human deletion in the human substitution in that same pocket, I'll demonstrate that where we are now sort of investigating. I will show you an in vivo preclinical model in mice, and then I'll show you a preclinical in vitro model that we are developing in the laboratory as well, using induced pluripotent stem cells. So there are some level setting things I want to use this slide to do. In the preclinical mouse model of disease, we're going to be comparing wild type and mutants. The mutant is that triple amino acid deletion. I will show you aging and a microbead model. And our endpoints are going to be intraocular pressure, optokinetic response, OCT analysis, and retinal ganglion cell counts to look at the structural and functional result of mutations in this particular functional pocket. Along same and in a somewhat orthogonal approach, I'm going to show you how reduction of this protein in our in vitro preclinical model demonstrates a similar neurodegenerative phenotype, but allows us to ask mechanistic questions in the future about why this is happening, how this is happening, and potentially the mechanism for which this can be reversed. And so these are, as promised, the visual alterations we see in the mice with the mutant WDR36 mutation. On the right-hand side, you're seeing IOP measurements in both wild type and mutant mice. Those IOPs, as the mice age, do not change without any perturbations. They're roughly the same. But alongside, as the IOP stays the same, we are seeing a reduction in OKR visual functional responses in the animals that are mutated for the WDR36. One of the highlights of working in the FM Kirby Center is having a resident specialist that can image these mice the same way every time and do that with minimal variability. And what we were able to find is that at 18 months, and we're collecting more information in older animals, that these mutant mice demonstrate a thinner retinal nerve fiber layer than their wild type littermate controls. Interestingly enough, and Bruce, when you mentioned rejecting the null hypothesis when the results don't necessarily tell you what you're expecting. This was an example of where we had tons of animals where we checked and counted retinal ganglion cells in the way that we count them. And despite reduction in the inner retinal layer, we didn't see any change in immunostained RGCs. And that was weird to us. One, we went back, we made sure it wasn't the assay, the way that we were counting the retinal ganglion cells. And so my hypothesis for this particular entity is perhaps there are subpopulations of retinal ganglion cells contributing to this inner retinal layer thinning that may be functionally relevant, but also may not be evidently picked up by just simply retinal ganglion cell counting. Additionally, we are still collecting data to count the nerve fiber layer. And so we want to count not just the soma, but the axons as well. When we challenged these animals with IOP using the microbead model, on the right-hand side, this is the pink and the blue are both the wild type and mutant animals that have elevated IOP. And if you look, those same pink and blue dots are demonstrating loss of visual function, but the loss of visual function happens a little quicker in the mutant WDR36 than would be expected. We also counted out those retinal ganglion cells, and that's an example of within as early as 2 weeks post-disease induction, we were able to see a reduction in retinal ganglion cells that had been stressed with elevated IOP. At 8 weeks, the damage was done, and as expected in the microbead model, we saw a reduction in both groups of retinal ganglion cells. And so our hypothesis is that there is a P53 related retinal ganglion cell loss that's happening in mutations in this functional pocket. I showed this diagram before. This is a blow up. What is believed to be mutations in the WDR protein elevate the amount of 18S ribosomal RNA that's present in cell cytoplasm. This particular signal is often used as a signal to activate the P53 pro-apoptotic response by inhibition of what is usually used in the cell to keep P53 activity in balance. And so when you see increased levels of 18S production in the cell, that is seen in some animal models of WDR36 insufficiency, you get activation of canonical transcription factors like BAX, B21, and NOXA in response to that particular mutation. And so with the help of a postdoc in the lab, we were able to take those animals, immunopan the retinal ganglion cells, and begin to look at some of the downstream regulators of P53 and demonstrate that in the mutant WDR36 mice, we were able to see activation of pro-apoptotic genes, more so than their littermate controls. And so what that demonstrates is that there's a natural severe loss of retinal function that precedes OCT structural changes in the mutant WDR mice. We see earlier, more severe vision loss and retinal ganglion cell loss associated with IOP elevation in the mutant WDR36 mice. And we think that this might be a readout of RGC vulnerability in the mutant WDR36 mice that may be occurring through the pro-apoptotic P53 pathway. As promised, we moved to a preclinical model of disease that included human cells. We like this because it allows us to evaluate the WDR36 genotype in a cell-specific way, but it also gives us direct human relevance. And so in this model, our goal is to use patient-derived iPS cells, reprogram them, and then do directed differentiation into RGCs to begin to tease out some of the mechanisms of this particular approach. The preliminary experiments I'm going to show is a knockdown of WDR36, and I will share with you some of the pathways of the lab moving forward so that this can be even more clinically relevant to some of the mutations that I showed you in the beginning of the talk. And so here's the data. In panel A, we were able to knock down about 50% of WDR36 response. Those cells were stained and measured for neurodegenerative phenotype. We measured soma size, neurite number, and neurite length, and we were able to demonstrate that the phenotype of those iPSCs is indeed showing smaller cells with potentially retraction of neurites that are less long and less able to send out dendritic projections. I should highlight that this work was largely done by my postdoc, who came as the trailing spouse of the Berger awardee for today. And I think that I am actually the lucky one because she accomplished much of this cell work. What this in vitro analysis demonstrates is that reduction of p53 causes a neurodegenerative phenotype in 2D culture. Reduction of this causes increased activation of p53 pathways during cell stress, and that mutation may cause increased RGC vulnerability to intrinsic stress. In this case, the cells just had knockdown of the protein. We did not expose them to any toxin. We did not stretch them or do any perturbation. They demonstrated the neurodegenerative phenotype alone. So I think that this is a great model for us to try to tease out what might be going on in the normal aging process. Some future directions are to continue to look at the aging and high IOP models of disease. My hypothesis is that what is similarly coming out is that there are subpopulations of cells that are affected and perturbed depending on the injury model that you use. And I think because it's the same gene mutation and we're seeing functional with no structural changes in the aging model, but more functional and structural changes in the IOP model, that this might be a nice model to use to delineate vulnerability of RGC populations. But for more human and more translatability, I am working in collaboration with the Quinn Lab to generate some of these mutations and knock-ins in iPS cells to further look at the phenotype, but also begin to ask questions about p53 pathway involvement that this mutation is causing. And so what this might do is fill a potential gap to delineate WDR36 as a potential genetic risk modifier. Patients carrying this diagnosis or this mutation might require earlier and more aggressive intervention. And for us, we are very, very interested in not just the mechanism of disease, but potential rescue experiments and further teasing out a potentially precise molecular mechanism. I want to acknowledge my lab. It's fun working with all of these individuals. I definitely do not do this work alone at all. It would not be but for the collaborative work of my entire lab, particularly changing media during the 2 very large blizzards we had here in Philadelphia to make sure that the cell work comes through. And so I want to thank everyone, and I am looking forward to questions.